Hi all, Dr. Clark here again. And for this lecture we're going to talk about the expression of phenotypes. So the previous lectures we talked about Mendel's work and his work with monohybrid, dihybrid, simple crosses, where the genetics is as simple as you have one gene from mom, one gene from dad, and the combination gives you the phenotype. Now, I want to talk about someone that really hardly anyone knows about. To tell you the truth, I bet you I could ask most of my colleagues and most biologists in this country who Barbara McClintock is, and they would not know. This is a travesty. Barbara McClintock should be known as the grandmother or mother of genetics. Her work paved the way for modern genetics. Yes, Mendel's work came before her. McClintock's work started in the 1900s, and around 1940 or so, 1930, 1940 was most of her work. She worked with corn and just did amazing research. And most of what I'm going to present to you today was the result of her research, that a lot of characteristics, I don't want to say most characteristics, but a good amount of characteristics that we currently know in organisms all over the planet do not fit Mendelian genetics. In other words, they are a combination of genes. So you can get what we call continuous variation, or you can get what we call incomplete dominance, or complete dominance, or epistasis, or environmental change. And it was McClintock's work that really drove this. She was the first person to describe what we call jumping genes. And these are genes that do not stay on chromosomes. She was also one of the very first people to present gene by gene interaction. And she probably deserves more credit than anyone for starting the fields of epistasis and epigenetics, etc. McClintock was also part of the result of hatred towards females in the 40s, and not individuals not giving females the credit that they deserved. Okay. McClintock worked on corn again. Like I said before, she discovered most of this research in the 40s. And she was ridiculed by fellow scientists. No one believed her except for maybe her own lab that she worked in. And she had a really hard time getting people to accept her research, even though it was really sound research. It was great. Now, one of the problems is she wasn't as good as some other individuals at maybe communicating her research. She was brilliant, first of all, and had hard time expressing very complex ideas into very simple form to people. But nonetheless, even people that understood her research didn't believe her. Okay? 40 years after her discovery, she finally won the Nobel Prize in 1980. And that really took a huge effort on her part to just keep, at it and keep people realizing that it was her work that paved the way for those discoveries. It was her work that did this. Okay, and so there's two books, A Feeling for the Organism and A Tangled Field. Either one are excellent. I highly suggest, if you're interested in the history of science or women in science, that you look up McClintock's work. She is amazing. It should be considered an idol to not just women but men, and a reason to not give up, even though you might have to wait 40 years to get recognized for the work that you did. In this case, it was well worth it. It's unfortunate because, if they recognized her work 40 years ago, we could have already started on genetic stuff that we're doing today, or maybe in the early 2000s, we could have figured it out in the 80s, maybe even the 70s, and it was because of her work that we could have done that. Okay, so with that being said, we're going to start looking at some examples of when traits don't fit Mendelian genetics. Okay, so phenotypes are not expressed as straightforward as most of the books present it. Okay, for example, almost every textbook in high school, and maybe even a lot of college textbooks, talk about eye color, and how eye color, blue eyes are recessive and brown eyes are dominant. This is all hogwash garbage, okay? Because your eye color is not controlled by a single variable. It's not controlled by a single gene. It's controlled by multiple genes. Okay? And so you have genes that lay down the pigment. You have genes that control where the pigment's placed. You have genes that lay another layer over the top. And so you can get all kinds of varieties. So it's not as simple as you blue eyes, you're recessive. Okay, that's not how it works, and that's why you see things skip. So like you can have two brown-eyed individuals, and they produce a blue-eyed baby, and the old joke is, oh, he's got blue eyes, must be the mailman or the milkman's baby. Well, no, it's because it's not a simple single gene trait like we present it to everyone, but that's not the way it is. So phenotype is often not expressed very straightforward. For example, we have what we call continuous variation, where you have multiple genes that influence a character. Okay? And so we call this polygenic. So you can inherit genes from grandpa's line, grandma's line, okay, great grandpa, great grandma, etc., and they build on each other, okay? And they influence a single character. One of the characters that's most well known for this is height. Okay, and this is why most people are not, halfway in between their mother and father. Some, if you're 3 children in a family, and dad's 6 foot, mom's 5'5", 3 children in the family, one could be 6'2", one could be 5'2", and one could be 5'9". Okay? It's because there's a lot of variables, a lot of genes that go into the height of individuals. Okay? So it's a continuous varying character. Another one, pleiotropic effects. So pleiotropic effects, okay, can be that you have one gene or one variation of that gene that can affect many different phenotypes. Okay? And so we often think of this one gene, one phenotype interaction, but you have to remember, we know that there's over 100,000 proteins, but there's only 25,000 genes. So we know that some of the genes, most likely, they have to code for more than one protein. Or there's a lot of proteins that are the combination of multiple genes. Well, the arrangement of those genes inside that protein will give you a different character, maybe a different phenotype of the same trait. Okay? So pleiotropic effects means you have one gene, but it has multiple effects on multiple different things. Cystic fibrosis and sickle cell anemia are examples of this. We know that cystic fibrosis is a lung disorder, okay? Overproduction of mucus. But things that we often don't talk about is what are the other effects of the CF gene. Okay, so the cystic fibrosis gene, when it's defective, when it's mutated, well, I shouldn't say it's mutated, when it's in its recessive form, cystic fibrosis is a recessive disorder. When it's in its recessive form, you overproduce mucus, okay, in the lungs. And this is the most common thing that we talk about with individuals who have cystic fibrosis. But we don't talk about the other things that go on. So sweat glands, so their sweat glands are altered, their pancreas is altered, their liver is altered. And so you have these genes that are controlling for a lot of other areas and have effects on those other areas, proteins in those other regions. Okay. Incomplete dominance. Okay. So this is interesting, and this is what Darwin was running into with his experiments on roses and other geraniums and things like that, is that not all flowers are simple as purple flower, white flower, nothing in between. Incomplete dominance is a character at which you show some of the dominant trait and some of the recessive trait. And in some cases, you show a little bit of each and you get a mix. And so maybe it's a mix of color, maybe it's a mix of height, maybe it's a mix of placement of the flower, a mix of hair on the fingers. You know, there's lots of different situations where you can kind of have a mix of the dominant and recessive trait. Right? And so this is one of the reasons, that Gregor Mendel's fourth hypothesis is now rejected, is because, or fifth hypothesis is now rejected, is because heterozygotes can have their own phenotype. They do not have to be dominant, like the dominant phenotype. They do not have to show dominancy. They can show their own phenotype. And this is where Darwin was struggling with his experiments, is because the plants that he chose showed incomplete dominance and codominance. So here you can see incomplete dominance. You can get red flowers mixed with white flowers, and the F1 generation gives you something completely different. So heterozygotes have pink flowers. They have a combination between the red and the white. Right? Now it doesn't have to be a perfect combination. It could be something that's even different. We know that if you mix red and white, you get pink, but you can have different. You could have just splotches of pink on a white flower. Or sometimes you get splotches of white on a red flower. And so incomplete dominance doesn't have to be this perfect mix. It's just something different. It's not really predictive. Okay? And sometimes the heterozygotes will have 2 varieties or 3 varieties or 4 varieties, which makes it very difficult to figure out, those test crosses and figure out what's dominant and what's recessive. Environmental effects. This is probably the biggest area of research currently today, is trying to determine what's the effect of the environment, or that the environment will have on alleles. Okay? Can the environment turn genes on? Can the environment turn genes off? Okay, and McClintock's work showed that indeed genes turn on and off, which was amazing when people finally read her stuff and believed her, to figure out, oh my goodness, she already knew this. She already found this in corn, that corn had jumping genes. They'd move. Corn could turn genes off and turn them back on, all kinds of things. So now, with environmental effects and with global climate change and things like that, we're now interested a great deal in how does the environment dictate what alleles are on and off, and the likelihood for those alleles to maybe move or combine. So maybe you get a continuous variable with environmental effects. We don't really know. Okay? But we do know that some genes are really heat sensitive. Okay, so if the temperature outside warms up, okay, you can get a gene turning on or off. Okay, so Arctic fox is a good example, but snowshoe hares, lots of things that have the ability to change their fur color between seasons have this going on. So as it warms up, okay, melanifers are turned on, the protein producing pigment is turned on, so that gene's turned on, and you get this pigment on the white fur. Okay? When it cools off, okay, those genes are turned off, and when the organism sheds its fur, it comes in all white instead of having a brown or black color to it. Okay? And so this is just environmental effects. And then we don't know all the environmental effects. We believe, scientists believe, that there's environmental effects with diet, with chemicals in the air, with chemicals in maybe possibly clothing or makeup, or chemicals you might be exposed to at your workplace. There's all kinds of situations that we're just not there yet. Now, there is some evidence out there that definitely suggests there are some environmental effects on alleles, and we know some of these, and they're well documented. But others, we're just beginning to test these kind of fields. Epistasis. Epistasis is when you have gene by gene interaction, okay? And you can have lots of genes interacting with each other. So again, eye color is an epistasis, is an example of epistasis. And so in some cases you have like two genes. One gene contributes something, and then another gene masks over that original contribution. Okay, so basically, epistasis is one gene modifies the phenotype or the expression of another. And there's lots of ways in which epistasis can work. You can have epistatic events where color changes, or where one gene turns on another gene, turns off another gene. There's lots of ways in which epistasis can work. And again, none of these fit Mendelian inheritance. And so if Mendel had picked an organism that had lots of events of epistasis, continued variation, okay, environmental condition changes, things like that, would have never found out his rules and his laws of inheritance. And even today, we're going back and making sure that we understand, like, most organisms don't follow Mendelian inheritance. Okay? Most organisms have some environment effect, or some gene by gene interaction, or some continuous variables. And there's lots of situations going on, especially complex organisms that have lots and lots of genes. You get a lot of interaction between those genes. Okay, so corn, and this comes from McClintock's work exactly. Corn is actually two pigments. So one gene that controls pigment being laid down, okay, and one gene that controls the production of pigment. And so you can get these differences in production of pigment in what we call wild corn. Okay? Most of you probably just recognize corn as being yellow in kernels, but they're actually blue or purplish in kernels, or red in kernels, and yellow and white, and there's a large variety of kernel colors in corn. And so her work showed epistasis in that. Okay? Another great example of this is, if you know something about things like Labrador retrievers, okay, this is how you can get some of the varieties of Labrador retrievers. Now, it's not just a two gene interaction. There's more genes than that interacting, okay? Because you have silver labs, you have charcoal labs, you have red labs, and these are just variations of these dogs. But for simplistics, okay, we can look at epistatic interactions for coat color in these dogs. So if you want a true yellow lab, or what sometimes is called a white lab, then it's recessive for both fur color and skin color, and will have a pink nose and white fur, or yellowish white fur. Okay. A yellow lab will have a dominant trait for skin, so it'll have a black nose and yellow fur. A chocolate lab will have dominant for fur color, but recessive for skin color. So it'll have a darker coat, but a pink nose, a light nose. And then a black lab will be dominant for both. Okay? And this is just simple epistatic interactions. And you can tell, that there's a difference in coat color. Right? Even though a chocolate lab has the dark or the dominant coat color, because of its skin color, because of that recessive gene, it produces a brown versus a black. Okay? And this is interaction by genes. Okay? Codominance. A gene will have often two alleles in a population, and so therefore you can have similar effects, okay, but two separate alleles. So sometimes this results in a heterozygote having its own characteristics. That's often the case. So it's not expressing either a recessive trait or a dominant trait. It expresses its own characters. Okay? And so they're often considered codominant. That means the recessive and the dominant are codominant when they come together. They both show characteristics, and it's unique to the heterozygote. Okay? Probably the best example of this is blood typing, or the one that's probably most useful for many of you is figuring out blood typing and figuring out antigens and antibodies from the different blood types. Okay? So you know that you can be you can have a blood type by being dominant for both. You can have a blood type by being dominant for one. Okay? And so, but if you're AB, that is your blood type. So if you have both the dominant and recessive trait, you can be AB. And so the heterozygote in this case carries its own blood type. Okay? And the recessive individual carries their own blood type. And so it's a very interesting situation. So you have four main blood types, and depending on what you inherit will depend on what your blood type is. And I'm going to show you a little video about blood types and the difference between A and AB and B and O here in a second.
Type 2 hypersensitivities involve interactions of antibodies and surface antigens of cells, followed by complement-assisted lysis of these cells. A typical example is mismatched blood transfusions. There are 4 different blood groups based on types of antigens on the surfaces of the red blood cells. People who are type A have A antigens, and those who are type B have B antigens on the surfaces of their red blood cells. Persons who are type AB have both A and B antigens, and those who are type O have neither A nor B antigens. The serum of people with type A blood contains antibodies against type B antigens, and the serum of people with type B blood contains antibodies against type A antigens. Type AB serum contains neither antibody, and O serum contains antibodies against both A and B antigens. If blood from a person who is type B is transfused into a person who is type A, antibodies present in the type A blood react with the surface antigens on the incoming red blood cells. This leads to complement fixation and lysis of these cells. If blood from a person who is type A is transfused into a person who is type B, antibodies present in the type B blood react with the surface antigens on the incoming red blood cells. This also leads to complement fixation and cell lysis. Persons who are type O lack A and B antigens on the surfaces of their red blood cells and are therefore universal donors. Persons with type AB blood lack antibodies against A or B antigens and are therefore universal recipients.
Okay, so there you can see a codominant situation, okay, in our blood system, okay, around antigen and antibody production for our red blood cell types. Okay? And I just want to just put it out there. You probably hear it a lot. If you're type O blood, please don't consider donating blood It's really important because you're a universal donor. There's always need for O blood, especially if someone who has O blood needs a transfusion or something like that. They're really the O blood can only be matched with O blood. So it's really important for people with O blood to give blood regularly. Please do so. This is just kind of a side note, but you can really help a lot of people's lives. All right, so with that, next time we come back, we're going to go into things like pedigrees and look at kind of more modern genetic research and modern genetic ways to look at DNA and components of genetic inheritance. Okay, next time.